Back in 2019, you know, the before times, I went on a trip to the Daintree and it was beautiful, teeming with wildlife. There were possums, birds, snakes, insects, and cane toads. Lots and lots of cane toads. If you're unfamiliar with the story of the cane toad, here's a summary. Cane sugar plantations in Queensland were being eaten by native grey-backed cane beetles and French's beetles. To get rid of the cane beetles, cane toads were imported from overseas and released, and then ate everything but the cane beetles, became a big old ecological menace, and now there's about 200 million of them hopping around causing problems everywhere. A classic tale of an invasive species, right? But how could the people releasing them have been so stupid? I mean, surely this was bound to happen. It's the same thing that happens every time an exotic species is introduced, right? The truth is, only a small proportion of introduced species become invasive. To better understand this, we can look at the rule of tens. Of all the species that are imported, 1 in 10 appear in the wild. Of those that appear in the wild, 1 in 10 become established, meaning they exist in a self-sustaining population. Of those that become established, 1 in 10 become invasive, meaning they cause harm to an ecosystem. And there are exceptions to this rule. Animals used in biological control, that is, animals introduced to control the population of another species, have a higher likelihood of becoming established, because a whole lot tend to be released all at once, a la Prince of Egypt. When people initially let cane toads loose in Queensland, they had past evidence that biological control was successful. In fact, one of the inspirations for the cane toad introduction was a story of biological control so successful we built monuments to it. In the 18th and 19th century, Opuntia, commonly called the prickly pear cactus, was introduced to Australia from Central and South America. They were intended to be used as food for cochineal insects, a bug that, when crushed up, creates a brilliant red dye, often used in food. In fact, even today, if you look at the ingredients on processed food, you might see carminic acid, cochineal, or 120, which are all code for crushed up bug dye. Yum! The prickly pear was also planted for agricultural fencing, but everything didn't go as planned. They quickly spread everywhere, choking up the land and making vast areas of agricultural property unusable. At its peak, the cactus covered an area of roughly 240,000 kilometres square, about the size of Great Britain. And this stuff was tough. They tried dousing it with chemicals, they tried burning it, they tried cutting away at it, but nothing could get rid of the prickly menace. In 1926, scientists tried something different and imported a humble little moth from South America named Cactoblastus cactorum, whose larvae love nothing more than to gobble up prickly pear cacti. I mean, the name says it all. Tell me Cactoblastus doesn't sound like a Pokemon. And as we all know, bug types are strong against grass types, so the Cactoblastus blasted the prickly pear out of existence. Seriously, the larvae did such a good job that some prickly pear species were almost wiped out in Australia entirely. So why did one introduced species fix the problem while the other made things worse? It comes down to a couple of factors. The research into which species would be best to control the prickly pear was pretty comprehensive. Scientists actually travelled to South and Central America where it naturally occurred to learn about the animals that eat it. Rather than picking a creature that may or may not eat the cactus, they wanted one that had evolved to do just that, so they could be confident the project would work. Even then, when they took them back to Australia, they conducted stringent breeding and feeding experiments to make sure they wouldn't attack other plants. With the cane toad, things played out a little differently. After hearing about the successful use of the cane toad in Puerto Rico at the 1932 World Conference of Sugar Technologists, scientists were inspired to import some to Australia. They were captured in Hawaii, where they're also an invasive species, and brought back to Australia to be bred. There was plenty of research done on how the cane toad interacts with the ecosystem as well, and there were concerns the toad would cause problems. The people releasing the toads weren't ignorant. At one point, the introduction process was even halted because of these concerns. But the beetles were still destroying the sugarcane, and in the end, it came down to choosing between economical or ecological loss. We all know how that story goes, so the introduction program was kicked back into gear. So you've got one introduced animal that's a specialist, eating a specific plant with no interest in any others, and one that's a generalist, preying on any animal it can fit in its mouth. Not only that, but the cane toad was a predator in an ecosystem where none of the prey had learned to avoid it. It had an Aussie buffet, reducing native wildlife populations and outcompeting other predators with its huge appetite. As a specialist, the Cactoblastis moth was primed to prey on its intended target. Prickly pear are CAM photosynthesizers. That means they open these little leaf pores called stomata at night to absorb CO2, storing it as malate, rather than opening their stomata during the day when they risk losing more water through the openings. Cactoblastis moths have this cool ability to sense CO2 gradients when they're flying around at night, meaning they could locate the prickly pear by sensing the dip in CO2 levels around it as it absorbed the molecules from the atmosphere. But the toad, while successful in controlling pest insect populations in Puerto Rico, isn't a natural predator of the Australian cane beetle. In fact, its lifestyle is so different to theirs that it rarely came into contact with them. The cane plantations are pretty exposed environments which the cane toad isn't keen on, and the beetles it was meant to eat sit high up on the cane where the toad can't reach. Like ships in the night, the biological control and its target just passed each other by, because, the toad could just hop away from the plantation and catch much easier prey. So, okay, the moth was more effective against the prickly pear because it actually evolved to eat it, but why didn't its population get out of control like the cane toads did? Again, this is because of how highly specialized the moth is. Remember, it doesn't like any other plants. In order to reproduce, it needs to lay its eggs on the cactus because its larvae need the cactus for food and shelter. Its specific environmental and food requirements meant that once the moth had done its job and the prickly pear population plummeted, the moth's own numbers followed suit. The two species are in this state of oscillation where when the cacti population increases, so does the moth, which causes the cacti numbers to go down and the moths follow. And as the moth population is reduced, the cacti can increase, and so on in that cycle. There are some species of Opuntia that don't host Cactoblastis larvae, and they're still a bit of a problem. Cane toads, on the other hand, are strong, independent generalists that don't rely that heavily on any other organism. If a prey population gets too low, they can just move on to something else. They're also able to tolerate all kinds of environments, even traveling across deserts by going from water body to water body. The population near the border of Western Australia has even evolved longer legs to travel faster from dam to dam. Damn, they're adaptable. And again, where the moth's ability to lay eggs is curbed by cactus numbers, toads aren't limited in that way. They just need any water body to lay their eggs in. Female toads can lay up to 30,000 eggs at a time, and their babies develop legs a lot sooner than most Australian native frogs. So the cane toads are able to leave the water earlier if they need to, giving them another advantage over Australian amphibians. So can't predators here eat them? Well, they can try, but it probably won't end well. You see, cane toads are toxic throughout every life stage, even from the time they're little eggs. These patches here are called parotid glands. They produce a compound called bufotoxin, which is deadly to predators that ingest it. They have other glands on their back, and their skin is also toxic, but this is the major problem area. The toxic meal they provide predators has decimated some native populations. Quolls have been especially hard hit. And there's another thing that makes them problematic: our attitude. People obviously like the moths, so if there was a news report saying that Cactoblastis had been spotted in the area, people would shrug. But when a news report says that cane toads have been sighted for the first time in a new area, people get pretty anxious. Surveys have found that the attitude of a local populace towards cane toads gets especially anxious, angry, and just generally negative when cane toads are first reported as entering into an area, and that anxiety then tapers off after a while. Now, this is a problem because when people are worried, they tend to act rashly. When the toads spread into new areas, people tend to take matters into their own hands and kill any toads they see to try and stem the flow. But not everyone is an amphibian expert, so this leads to the unintentional killings of native Australian frogs that get mistaken for cane toads. Today, there are a couple of things being done to control the cane toad population, including toad-proof fencing in Western Australia, genetic experiments to release sterile toads into the population, and even some pretty successful experiments to condition native predators like quolls to avoid toad meat. They do this by microdosing the quolls on the toxin to make them associate the meat with feeling sick, and then they release them hoping they'll pass on this avoidance to their offspring. Some animals have even independently learned to avoid the poison glands by flipping the toads on their backs to eat them. So there you go, sometimes introducing a species for pest management works, and sometimes it doesn't. When it goes well, it's good, and when it doesn't, it's really bad. So at the very least, it's probably best we don't introduce anything else to control the toads.